here, if you see that the major cells of innate immunity, I already, why it's showing here, 1 minute. So I was here actually in this slide, I already explained this. Now here, the major cells of innate immunity, as I told you, like neutrophil and natural killer cells, they also take part in the innate immune system, but at this level it will be difficult for us to discuss, so just forget about these two. But we cannot ignore this macrophage and dendritic cell. Okay, we have to understand. So in order to discuss the further other thing, we need to know about these two. Macrophage and dendritic cell, both of them, they have properties phagocytosis. Someone of you actually already told me a few minutes back. The phagocytosis, that means they can take some foreign particle, it can be virus, bacteria, anything, and then they process that information and then they finally present the antigen. Okay, why I put phagocytosis here in top and here in down? Because the major function of macrophage is phagocytosis and also it presents antigen. But here in this case, its antigen presentation is the major function. Both are important. Like without phagocytosis, it cannot present antigen, right? So both are related function actually. Now, these cells are called big eaters. They are always hungry. That means actually, particularly this dendritic cell, they are they are called sentinel. That means they are actually, traveling through our different tissue, different portion, and as a result of that, what happened? Those whenever they encounter some pathogen, then they will start, reacting against that. So this is their job. They are big eaters. So they just, eat and digest and kill those pathogen, right? Then here you can see, I'm just giving you some particular scenario. Here, if you consider this is skin layer, and for some reason, some break happened, either small cut or some scratch, something happened. And some bacteria, they are going inside your body through the skin, the broken skin. And then whatever the bacteria enter inside, then in the meantime, some cells already damaged here, like in the broken layer of skin. From those cells, some components, they are getting secreted. Okay, so those are called, this is group name, like I would say not specifically, those types of compounds are called alarmin. They are providing some kind of alarm. They are called alarmins. It can be different types of component. You don't need to remember, but what I'm trying to say, this kind of mechanism exists in our body. So from those damaged cell, not from bacteria, remember, not from pathogen, from our own tissue, which are getting damaged, from those cells, some kind of compound, it can be protein, mostly those are proteins, they are called alarmin. So that alarmin, it can be, dendritic cell can sense this information that alarmins are around. So as a result of that, dendritic cell present somewhere here, it will travel towards alarmin. More alarmin present, the dendritic cell will go in that location. That means something happened there, some problem happened there. So now dendritic cell is coming there to fix the problem. What is the problem? Bacteria coming inside because of this wound, because of this broken skin. So the job of this dendritic cell is to, engulf this, eat this bacteria and kill that. Okay.
So
now,
alarmins are secreted by the epithelial cells or the endothelium cells, like which
mostly,
layer of skin?
Mostly your question is right, because since I told skin, that's why. So as far as I know that endothelial cells definitely it is getting secreted, but epithelial cell I am not very much sure, but mostly it can be like epithelial cell that doesn't mean skin. Even epithelial cell present on lung also, like alveolus, those are also epithelial cells. So as a result of that, both can secrete these alarmin kind of molecules. Like again, those are not specific molecule. Its name is alarmin, not like that. They are group of molecules. And now, whenever like I'm just giving you some very basic example like broken skin or wound, but it can be epithelial cell like we shouldn't consider that way. Sometimes we believe that epithelial cell means the upper layer of our body, that means skin. Not that is not the true information. Like even in like as I told you in the lung, that lung layer, the alveolus, those are also epithelial cells, right? So as a result of that, whenever those dendritic cells senses this molecule, they will travel and they will try to destroy this bacteria or pathogen. But the thing is, the pathogen will also increase in number. So then it will send or process this information to our adaptive immune system. So here again we are showing the same similar kind of thing. If the epithelial barrier, in this case it is skin, what happens after a breach? So for example, if you see this is the broken portion of the skin, some bacteria going inside. This is I have taken from textbook. So this is macrophage, this is dendritic cell. They are actually destroying those pathogen or bacteria in this case. And now what happened while they are doing this, then you will see some kind of symptoms. Say I'm coming back here. So what happened if you have small infection, small cut, something happens, small infection, then what you normally see in that area? If I ask you symptoms, I think most of you can, you will be able to say that little bit of redness, little bit of swelling, right? Sometimes it's like pain. Sometimes after little bit of longer time, some, pus formation. Those are common sign of infection, local infection. All of you encountered this problem, right? So why those things happen? Because of this battle. Those things are not bad actually, those are good. That means your immune system are actually functioning, they are taking care of those things. That little bit of also at the initial phase, the local area getting little bit warm, like little the temperature rise little bit in locally. So those all those things are good sign. So now if you see here, I'm just showing you what happened. So a fundamental type of response by the body to injury or local infection is characterized by the classical sign of vasodilation. So vasodilation means like in that area, I should explain. So something happened here. So in this region, we have blood vessel. So blood vessel get dilated. That is called vasodilation. Why? Because this region now needs lot of macrophage and dendritic cell, because in the tissue in the surrounding area you have some macrophage, some dendritic cell, but you need more. Then how those will come? Those will come from different parts of our body. How they will come from different parts of our body? They will travel through cells and tissue? No, that will take longer time. They will come through blood. Okay, so blood flowing very quickly in this region, and blood carry those dendritic cell macrophage also, right? And blood flowing very quickly in this region. That's why it's little bit warm also, little redness, as I told you. Local infection at the beginning, little bit of redness. And more and more immune cells, they are coming in this local area, in this infected area. And the battle is already started. And since the blood vessels, they get dilated, that means vasodilation, whatever the term I told. Some fluid will also be leaked, like from the blood vessel, like some fluid, whatever present, the fluid inside the blood. So as a result of that, that portion will be soiled little bit. That's why swelling happens. All those things are classical symptoms of local infection and injury. And that is not bad at all, as I told you. And finally, everything will get cured after a few
So
days.
can you repeat the mechanism? I kind of got lost.
Yeah, so in the slide, you will get everything. I'm just telling you again. So if some local infection happen, some cut or some injury, some pathogen then come. Now, in this area, like some tissue means some cells, like cells present here inside, like there, some of those dendritic cell macrophages, they are actually traveling and they are sensing those information. Now they are not enough to clear this infection. They will send some signal. I will send you one video link. You will get a nice video of around 8 minutes. You will get all these things. So they will process the information and then they will recruit more and more dendritic cell macrophages. How they will come? That's why I'm telling that if the vasodilation, that means blood vessel get dilated, then cell will come out from the blood vessel to this tissue. Cell means what kind of cell? This dendritic cell, macrophage. So that's why they are like different, like those kind of cell. They can even escape from narrow space. When the blood vessel get dilated, they can escape from the blood vessel and they are coming in this region and they are taking part here. During that time, little bit of fluid get leaked from the blood vessel. That's why swelling happen. And finally, it can turn into pus formation and all those things. But finally, if you see, all those things are happening because of a nice battle between our immune system and pathogen. Now, if that is not enough, what will happen when this pathogen, like this macrophage and dendritic cell, they are actually destroying this pathogen or bacteria. So they process this information and then they present this information to our like adaptive immune cell, that is called T cell. And T cell understand what kind of pathogen there, and based on that, T cell tells B cell, okay, make antibody against this cell, this pathogen, and those antibody can directly target those pathogen and kill those pathogen. I'm sure that last 1 minute, whatever I told you, have multiple questions, but you will get answer within 15-20 minutes while I am going through it. So now. I'm not going through it anymore, so I already explained this thing. So again, this slide, because what I was trying to say, whatever I told, those things are happening somewhere here, somewhere here. These innate cells, they are fighting, and at the same time, the adaptive immune system will slowly learn how to, how to make the antibody against, or like what kind of antibody we should make to clear this infection. So here in this case, the point we should discuss here, the transition from innate to adaptive immune response. Because after certain time, if your adaptive immune system is not taking care of this thing, innate system will not be able to take care of all kind of infection. So as a result of that, adaptive immune system should play some role here. But how this transition will happen? Here, very clearly I have written here, this is important, that very slowly I'm going also, just try to understand. Proteins eaten by APC. APC means antigen presenting cell. Antigen presenting cell means here antigen presenting cell, APC. Who are antigen presenting cell? Macrophage and dendritic cell. How they will present antigen? Because they engulf those pathogen, they destroy those pathogen, and that is what we are talking about. Proteins eaten by APCs, not proteins. That means whole the pathogen, right? Whole pathogen, and inside the pathogen, that means in bacteria you have many proteins. So that proteins we are talking about, the foreign proteins. Proteins eaten by APC, antigen presenting cells, are broken down to small pieces, where inside those antigen presenting cell. And small piece of protein should be what? Peptides, that you know in the protein class I told, peptide, right? So small pieces of protein, that is peptide, which are loaded on special receptor Those are called MHC. I will discuss little more details later. MHC and transported to the cell surface. Peptide-loaded MHC complex can be recognized by T cells, and that interaction can lead to adaptive immune response. Like here, everything summarized about adaptive immune system. Let me go just see or listen. I will very like basic way I will discuss this thing. What happened? So this is antigen presenting cell. Okay, it can be dendritic cell, it can be macrophage. What happened when they eat some bacteria? So this dendritic cell or macrophage, they have the enzymatic machinery. They will finally destroy those pathogens. They will clip into small pieces. Okay. And this is one protein, this is another protein, this is one carbohydrate coming from the bacteria. That's small fragment. What happened? Some of those peptides coming from this pathogen, those peptides are called antigenic peptide. Antigenic peptide, not our own peptide. See, antigen presenting cell, that means they present antigen. Dendritic cell and macrophage, they are our own cells. They are our own body cells, immune cells. But these pathogen are not our own cell, right? They are pathogen. They are intruders. So what happened after this breakdown? Some of the foreign peptides, that is called antigenic peptides, are displayed on the surface of these cells. Okay, so they have some molecule. Okay, this molecule present on the surface of APC. These molecules are called MHC, major histocompatibility complex. Those are some protein receptor present on antigen presenting cell. And what they present? They present antigen. Okay, this antigen are foreign, right? This antigen are coming from where? Tell me quickly. Let's unmute. This antigen coming from where? Any one of you, please. These antigens are coming from where?
From the peptides of the antigens.
Yeah, you can say loudly. You're right. They are these antigen are actually part of this pathogen. When antigen presenting cell, they destroy those things. Some of those peptides, they are presented by those molecule receptor present on the cell surface. Those are called MHC, major histocompatibility complex, and this is your antigenic peptide. Okay, antigenic. Sorry, is yeah, antigen or antigenic peptide.
So what was the full form of MHC you said?
Yeah, so that's present in the slide. Okay, major histocompatibility complex. Okay, just remember as MHC. But this is very important. The MHC, they only present, antigen in our body. No other this is very important molecule. They only present, this antigen. And as a result of that, now what is the mechanism? Now we have T cell in our body. Okay, so this is called T cell. My goodness, T cell. So T cell and B cell, they are part of our adaptive immune system. Now T cell, they have some specific cell surface receptor that will interact with this antigen. That means T cell is trying to understand what type of antigen present, because this antigen is actually part of this bacteria, part of this pathogen. And as a result of that, this T cell, while it is actually observing this antigen, this T cell can take, it can work in two different ways. One way, this T cell will actually give some signal to B cell, okay, make antibody against this kind of, again, this peptide. How? That is very complex thing. I'm not going into that details, little bit present in the slide. And then this B cell will make lot of antibody, okay, lot of antibodies. Those antibodies can react, can bind to this peptide. Okay, so as a result of that, what happened? Those peptides are coming from this bacteria. So as a result of that, when those bacteria are present in our body in huge number, but B cell produces antibody, antibody will directly bind to those bacteria and mark those bacteria, and that will help our immune system to clear that infection. So that way antibody helps to clear infection. And the T cell, it has two functions. Either it will teach our B cell how to make antibody or what kind of antibody will make, or the T cell will be reactive. This is called cytotoxic T cell. Cytotoxic means, cyto means cell, toxic means toxic, you understand. So this T cell will directly kill the infected cell. So as a result of that, this T cell, either it will teach the B cell that to make antibody, or this T cell will directly, kill the infected cell. So as a result of that, both way they will clear the infection. So now you can ask me that why T cell are doing these two different jobs, because we have two different types of T cell. One is called helper T cell. So helper T cell, actually when see antigen, they actually help B cell to make antibody. And the other T cell is called cytotoxic T cell. That T cell actually itself, it kill the infected cell. That way, finally, adaptive immune system help to clear the infection. Okay, so let me go through the slide. You will understand. So those things I'm not going into that. I already discussed. So here, little bit of information present here. I'm coming little later because otherwise, whatever I told that part, I need to explain you first. Here, dendritic cell. That means this is actually APC, antigen presenting cell, right? So it has MHC on the surface, and when that MHC present antigen, we have two types of T cell, as I already told. Those T cell will identify the antigen presented by this MHC, and on the basis of that two pathway. In one pathway, you don't need to know the details of that. In one pathway, it will make antibody, and the other pathway, it will clearly kill it will directly kill the infected cell. So mostly we have two types of T cell. One is called CD8 T cell. CD8 is a protein name. Based on that, it has been named. So that CD8 T cell directly kill infected cell, and CD4 T cell, they teach our B cell. B cell is another population of our adaptive immune system to understand and teach how to make antibody. Okay, so this is the overall mechanism. Details you don't need to go through it. I will rather I will not discuss in details. I will give you time so that you can ask me question, and I would like to clarify your answer. Okay, so let me explain this part also. Immune cells and the lymphoid organ. So this is human body, right? And you can see here that this is bone marrow, here bone marrow, right? I told you that all blood cells, all immune cells, they are actually coming from bone marrow. It has stem cells, and bone marrow, from bone marrow we get all sort of different types of cells, right? Now the point is, this red color organ, bone marrow and thymus here, thymus, those are primary lymphoid organ. That means they make these immune cells, right? Even in thymus, little bit of important function happen. I will tell you now. And this blue color thing, they are the secondary lymphoid organ. That means they store those immune cells. White blood cell. White blood cell means those, eosinophil, basophil, neutrophil, those are actually T cell, B cell, those are white blood cell. Don't forget about this thing. Red blood cell means RBC, which carry oxygen. They are not taking part in our immune system in that way. White blood cell means those immune cells. They don't have hemoglobin. That's why they are called white blood cell. They are not red blood cell. And that means all these immune cells, they are part of white blood cell, WBC. Okay. Now, they are stored in this secondary lymphoid organ. Another important thing is, as I told you, the bone marrow contains blood forming stem cells and make B cells and other immune cells also, like innate cells and other blood cell type. Like that means B cells are coming from bone marrow. But what about T cells? B cells and T cells, they take part in adaptive immune system. T cells are also initially produced from bone marrow, but they are not functional. Those T cells will come to thymus, one small organ present here that is called thymus. This is also primary lymphoid organ because those immature T cells, they get mature in thymus, and now you have T cells and B cells, they can take part in our adaptive immune system. I would also like to mention one important thing here. See those secondary lymphoid organ present here. Probably see that because I will show you something here. Also some secondary lymphoid organ here. You cannot see here right now, but here you can see. So now what happened? This is important actually. When someone is suffering from fever for a long time, if you go to doctor or some other disease, what they do? They test whether these glands are swollen or not. I'm sure that you have seen that here gland, right? Why? Because those are actually secondary lymphoid organ. They store, those immune cells. So when someone is suffering, it is expected that those glands will be swelled because they reserve those. They are actually, making more and more immune cells, and they are stored here. Not only here, there are many other locations actually. That's why I was showing in that figure. Okay, so if those glands are swollen for few days during this infection and disease, that is good again. That is not bad. Okay, so that is actually whenever lot of immune cells are getting generated and they are getting stored, and they are taking part in this battle also, right? So that way they actually help. So now if we go back here again, so all these things, that antigen presenting cell, major histocompatibility complex, I discussed already. Only thing I would like to mention here, this MHC, this is so important about, like, see how many Nobel Prize you can see: 2011, 96, 87, 84, 80, all related to this MHC and its interaction to T cell and the production of antibody. Like this is actually core of our immune system, or this is the major thing. That's why we are protected, and that many Nobel Prize they just received it because that their contribution regarding MHC, T cell, and B cell. Okay. And this part I just draw in the whiteboard and I tried to explain you. And then what are the antibody? Antibody, they are getting secreted from B cells, right? So in this slide you will see that B cell, and after that you have plasma cell. So antibody secreting B cells, they are called plasma cells. Okay, the B cell which secrete antibody, those are called plasma cells. These plasma cells also nowadays, if you are going thoroughly through newspaper, you will also get this plasma cell, B cells, antibody secretion, all those things even in newspaper. Okay, so plasma cells, they secrete antibody. So what antibody looks like? Antibody is nothing but a complex protein. This is quaternary structure of a protein. And here you have antigen binding region here in this region. And here you can see, depending on what kind of group present here, what kind of, the shape and size of this antibody, based on that, it will recognize the antigen. As I told you that every time our, someone is getting infected, during that time, our immune system, they make weapon. Weapon means here antibody. I told weapon several times, but weapon means antibody. So that specific antibody. So adaptive immune system is specific. So those antibody will be directed towards only those pathogen, because that pathogen, the information of that pathogen has been processed by dendritic cell and macrophage, right? And then it has been presented through to T cell, and T cell finally, I would say help B cell to make antibody. So those antibody are basically directed to those pathogen. So as a result of that, when you have a lot of plasma cell, they will make huge amount of antibody. Those antibody will travel and mark those pathogen. That means pathogen means bacteria. Some of those protein are present on the surface of the bacteria, and that protein itself presented, that protein get digested inside the antigen presenting cell. They make small peptide, and on the basis of that peptide, the antibody actually produced. So as a result of that, those antibody will mark those bacteria or pathogen. And if whenever those antibody will mark those pathogen, then our other cells, they can quickly kill those pathogen, like kill those bacteria and other cells. So as a result of that way, both innate and adaptive immune system, they work together, and then they finally clear the infection. As I told you, I will send you one nice video, actually, a very important video. Just if you can manage your time, you should watch this video by today, so that whatever I discuss, you will get indeed, you will get the point. So before I will finish by 10:15, and I will discuss about the vaccination. But before that, I would like to answer a few of your questions.
Hello, sir.
Hmm.
So the B cells produce the antibodies from the information that was presented by the MHC and all. So and you said that the protein signature, according to that, it produces the antibodies.
Hmm.
So for different pathogens, different proteins will be there. So different antibodies need to be made. So and the proteins are made from processed from mRNA, we know. So does that it has to change its mRNA or something like that, or
So
how does it?
Before mRNA, whatever you told, that is nice summary. You understand properly. This mRNA part is not required. I'm just telling you. Until that, like before that, whatever you told, absolutely right, and you nicely summarize it. That different pathogen, they contain different protein also, right? Because they have different genome, they are different pathogen. They have different proteins also. Now, whenever some different bacteria they infect us, right, or some other pathogen infect us, our immune system respond and make antibody or weapon against that pathogen. Okay, so as a result of that, that can identify those pathogen, and our immune system can quickly clear that thing. But those proteins are coming from definitely from the mRNA of that bacteria, of that pathogen. But directly in this system, whatever we are talking, mRNA of those pathogen had no role here. They are making protein in those bacteria, and those protein are getting processed by our immune system. Okay, but now here I forgot to mention something like I would like to mention here. After the battle is over, like when the battle is over, like infection is cleared, some of these B cells will remain in our body for lifelong, like 10 years, 20 years, 30 years. Those cells are called memory B cells. Next time, if we get the same pathogen, same disease, like if we are infected by the same pathogen, this memory cell, they don't wait like all these like steps what I have actually discussed. Before that, they will quickly make antibody against that pathogen, because it has the information that how to make this antibody or what antibody is required. This is called memory B cells. So that's why for the next time, because of the exact same pathogen, most likely you will not get the disease. In rare cases, you might get disease, because as I told you, we are living system, we are not going through some kind of theory. This is some kind of always changing. So some because of some other issues you might get, but vaccination relying on this thing only, that if you can teach your immune system what is the pathogen, so that next time it will protect you, right? So now I will discuss about vaccination.
Excuse me, sir. Just a small doubt.
Hmm.
Sir, how does the body know? Sorry, means the B cell is there. No? So how does it know that the same the same pathogen has come? Because first T
Yeah,
cell
yeah,
is
very good question, Rajesh. Very good question. I didn't expect this actually, because the thing is, so simplified way I actually explained. I didn't. That's why I didn't expected this question. But if I get like 2, 3 more classes, I would like to bring those issues also. So here, Rajesh, to all of you, it's a very nice question. Actually, Basel, they also have an ASC. Okay, so they can also recognize the pathogen at the first place, but they cannot make those antibody. They need more time, right? That part already learned. So as a result of that B cell, right? Apart, I would like to mention you to all of you. Apart from red blood cell, all of our cells they have MHC. Although professional antigen presenting cells, they are taking the major part, but apart from red blood cell, all cells in our body, they have MHC. Okay, so even B cell, they have MHC. Rajesh, I think you got now. So those B cells, they have MHC. They can quickly bind to, the bacteria itself. Now, after binding, they can remember that the same thing they produce long time back, right? So as a result of that, that is the, I'm not going into like a lot of details there, but this is the idea how memory vessel can quickly identify, or the whole system can help to memory vessel to expand to plasma cells. That means antibody secreting B cells, and it can be,
So, one
yeah.
question. So, these B cells, which are memory B cells, suppose towards dengue virus, it is producing antibodies.
Huh.
Then will like they are specific to, they are made specific to that virus or pathogen only, or they can like neutralize other, like they can form
Yeah,
antibodies for other
this is
pathogens.
Tanmay, I got your question. This is good question. But the thing is here now it's rely on that it depends on those two pathogen or how closely they are similar. Sometimes two pathogen, many of their proteins are very similar. So as a result of that, this antibody can act against both the pathogen. So that way it is cross-reactive, like cowpox and smallpox, as I started the class. So all of you, like Tanmay and whoever asking question, let me finish little bit, because some other student, they might leave after like 10:15, as I told you. So after that, if you have question, again, we will discuss after 10:15. So just I have like another 7 to 8 minutes. We should little bit talk about vaccination, and particularly this Covaxin and Covishield, as I told you before. Let us discuss. Okay, then I'll be more than happy to discuss with you. So here, vaccination. So here, this is just cartoon. You can see that Robbins and Batman. So vaccinated kid, and this is unvaccinated, right? So the concept of drug is, kill invaders or foreign pathogen. Drug, like for example, antibiotics, inhibit the growth of pathogen. And the concept of vaccine is train our immune system to face various existing disease-causing agent and generate memory against the specific pathogen. So the next time, if someone get the same pathogen, so that it can identify and it can clear the infection without the disease. Okay, this is the difference between drug and vaccine. So here, immunological memory versus vaccination. So I actually explain all those things. So in case of natural infection, what happened? The first infection, that means someone get the disease, the slow response, pathogen multiply in our body, some symptoms happen, that's why we get the disease. And over the time, our adaptive immune system actually take care of it and clear the infection, and we generate our some immunological memory should be there. During second infection, few years later, the response is fast, pathogens dispose. As a result of that, no symptoms or no disease. This is the natural infection. We are trying to make it through vaccination. That means no disease. That means we are actually bypassing. The vaccination protect us from infection by inducing the adaptive immune response by bypassing the need for the disease. So here you can see, first, the vaccination means we are giving some inactivated form of pathogen so that the pathogen cannot multiply or it cannot create the disease in host. Rather, it will only activate our immune system, not activate, I would say. It will teach our immune system to make the weapon or how to make the weapon, and the memory cells will be there, and in future when the natural infection will happen, it will be similar. Similarly, it will respond very fast, pathogen will be disposed and no disease will be there. So this way vaccination work, right? So now let's just one small slide about success of polio vaccine. All of you know that polio is not a very severe disease, I would say. In some cases, it is causing problem. You can see here that this kid actually it has severe poliomyelitis symptoms. It also attack our nervous system. These legs cannot work properly. But most cases, it is not very severe. But the thing is, polio vaccine actually worked very nicely, and polio, I think that soon it will be declared that polio has been eradicated from the world actually. So just like smallpox, as I told you, right? So as a result of that, after that, no need to take polio vaccine again. So in 2012, only 3 countries, one in Africa and Pakistan, Afghanistan, they reported polio. And after that, I heard that because of some laboratory issues, actually some polio cases also detected in some parts of the world. But it is under complete control, and I believe that who will declare that polio should be has been eradicated soon. So because it's not there anymore. So now, if you see here, I would like to discuss about little bit about Covaxin and Covishield. Lot of information, not lot, just one slide here, but you can read this slide later. I will like to discuss first. What is the, you understand how our immune system works to some extent. Also, little bit you heard about what is vaccine and how it works. Now, this Covaxin and Covishield. First, let me start with Covaxin. So this Covaxin, vaccine which is generated in Bharat Biotech. So this Covaxin is a full form of virus. That means that this is the inactivated SARS-CoV-2 virus. I'm not going into the details of how inactivation happened, all those things, but this is the whole virus, but inactivated form, so that virus, after taking the vaccine, that virus cannot grow in our body. But what will happen? I just told you that virus will be ingested by our immune system and process the information, present the signature peptide, and teach our immune system how to tackle this thing, right? So as a result of that, our immune system will know how to tackle this virus. So that is how this Covaxin works. This, I would say, the way this Covaxin has been made, this is the routine vaccination, vaccine, routinely vaccines are produced in this way. Like most of the other vaccine, for example, rubella vaccine, many other, I mentioned here. If you see, like rabies vaccine, vaccine against seasonal influenza, polio, pertussis, Japanese encephalitis. So all those vaccines are actually made in the same way, that inactivated form of virus. Because if you remember, when last week I discussed about the viruses. Virus, they have capsid protein. They have many different types of protein. So in this case, when you are giving the full inactivated virus, so during that time, your immune system can see all those different types of proteins, and they can make process this information. But in case of Covishield, this is also like Covishield, and also other vaccines are present in the same strategy. And this is only they made it for this SARS-CoV-2. So Covishield, what happened? They use only spike protein encoding gene. Like apart from spike protein, they have some other protein also in SARS-CoV-2. But they took the information that make spike protein, and that information they put it in some adenovirus that is called chimpanzee adenovirus. Some adenovirus that cause some common cold in chimpanzee. So in case of Covishield, I'm talking about. So they put that genetic information that makes spike protein in that adenovirus. Which adenovirus? That causes cold in chimpanzee. That is some kind of system like adenoviral system. So now that is now a vaccine. So you are actually administering that in Covishield those adenovirus present. But those adenovirus are also incompetent. They cannot multiply. The virus cannot multiply, but they will go inside our cell and they will, whatever the genome present in the adenovirus now, they also contain the information to make spike protein. Whatever I taught you from the beginning, like replication, transcription, translation, all this information now required to understand this. Now, the information present in adenoviral genome that will go inside our nucleus where it has been administered, like when you are taking the vaccine. Now, inside the nucleus, those gene that will encode this spike protein that will be translated into mRNA, and from there you will get the spike protein. This is a foreign protein. Finally, your immune system will process this protein and finally they will make antibody against the spike protein. So what's the difference? In case of Covishield, you will get antibody only against the spike protein. Maybe the amount of antibody will be more because only it know how to make antibody against spike protein. But in case of Covaxin or any other traditional vaccine, there we are trying to teach our immune system to make antibody against the whole thing, whatever present, whatever the signature present on that particular pathogen. So this is the difference between, like as I told you before, I will discuss about the basic difference, but